Hello everyone, welcome back to the channel Microbial World. Today we will be discussing about transmission electron microscope. So before we move into the topic, let's see a brief introduction about electron microscope. Electron microscopy involves the study of different specimens using an electron microscope. And these electron microscopes, they are scientific instruments that use a beam of energetic electrons. Okay, they use a beam of energetic electrons to examine the objects on a very fine scale. And also the electron microscopes are having greater magnification than light microscope. Types of electron microscope. So there is transmission electron microscope TEM and scanning electron microscope SEM. So the transmission electron microscope, they are forming images using the electrons that is transmitted through the specimen. Okay, so you can see here an electron beam, it is incident on the specimen, and those electrons which is transmitted through that specimen is used to form image in case of TEM. And in case of scanning electron microscope, they are utilizing the electrons that bounce off from the surface of the specimen. Okay, an electron beam is incident on the specimen, and those electrons which bounce off from the surface of the specimen is used to form image in case of SEM. Transmission electron microscope TEM. So the transmission electron microscope use a beam of electrons to focus on the specimen, and they produce a highly magnified and detailed image of the specimen. Okay. The first electron microscope was developed by Ernst Ruska and Max Knowles in 1931. Later on, it was modified into transmission electron microscope by Ernst Ruska and along with Siemens company in 1933. So the magnification power of transmission electron microscope is over 2 million times better than that of light microscope. Okay. And how the image is formed in TEM? The image, it is formed from the interaction of electrons transmitted through the specimen. Okay. So when the electrons transmit through the specimen, these electrons, it will interact with the specimen and form an image. Okay. And that image is magnified and it is focused onto an imaging device such as fluorescent screen or a layer of photographic film. So now let's see the parts of transmission electron microscope. So the parts include electron gun, electromagnetic lens system, image producing system and image recording system. So this is somewhat how the electron gun looks like. Okay. So the cathode, it is a tungsten filament. You can see the tungsten filament here, and the tungsten filament, it will emit electrons when it is heated. Then there is a negative cap, that is the Wenelt cap. So this negative cap, it will confine the electrons into a beam. You can see the electron beam here. Okay, and this is the anode plate. So the electrons, it is transmitted from the cathode to the anode at high voltage. Okay. Then there is the electromagnetic lens system. So the electrons, it cannot pass through glass lens. So electromagnets called magnetic lens are used in transmission electron microscope. And its function is to focus the electron beam onto the specimen. Okay. Then there is image producing system, and this includes a movable stage to hold the specimen. Then there is an objective lens, projector lens. Okay. And its function, they are functioning by focusing the passing electrons through the specimen and producing a highly magnified image. Then there is image recording system. Image recording system consists of fluorescent screen, that is to view the image. Then there is digital camera to permanently record that image. Okay, so these are the parts of transmission electron microscope. Now let's see the working principle of transmission electron microscope. So as we said before, the heated tungsten filament in the electron gun, it will emit electrons. Okay, and these electrons, it is focused onto the specimen by the condenser lens. And the specimen, it will scatter some electrons, and only the electrons that pass through, that is only the electrons that is transmitted, is used to form image. Now consider, now see, now this is the specimen, okay, this is the specimen. Now a dense region of the specimen, it will scatter more electrons, okay, it will scatter more electrons, and only few electrons will be transmitted through the specimen, okay. So this region, it will form a dark image, and this is called electron dense region, okay, it is called electron dense region. Whereas a thin region of the specimen, it will scatter only few electrons, it will scatter only few electrons, and more electrons will be transmitted through that region, okay. So the image will be bright, the image will be bright, and it is called electron transparent region, okay, it is called electron transparent region, okay. So the objective lens, it is of short focal length, it is about 1 to 5 mm, okay. So the objective lens, it will form an intermediate image. The projector lens, it will further magnify the image, and the image, it is formed on the fluorescent screen or photographic film. Another thing that you need to notice, that all these lens and specimen, it is all placed under high vacuum, okay, it is placed under high vacuum. That is because the electrons, it can collide with the air molecules, and the electrons can get deflected. So these lens and specimen, it is placed in high vacuum in order to get a clear image. And we are using very thin specimen so that we can accurately and clearly visualize the image. And the image that is formed, it is called monochromatic image, which is grayish or black and white. So this is the working principle of transmission electron microscope. Advantages and disadvantages of transmission electron microscope, okay. So the transmission electron microscope is having very powerful magnification, okay. We said that before, they are having magnification of over 2 million times better than that of light microscope. And also using the TEM, we can acquire information on compounds and their structures. And also they produce very efficient high quality images with high clarity. And they can also produce permanent images. And another thing is, it is very easy to train and use transmission electron microscope, okay. The disadvantage is that it is very expensive to purchase, and also they are very big to handle and difficult to maintain. And they are extremely sensitive, okay. They are sensitive to the vibration and electromagnetic movements. So they are used in isolated areas where they are not exposed. And also the transmission electron microscope, it requires a constant inflow of voltage in order to operate. Applications of transmission electron microscope are, it is used to visualize and study cell structures of bacteria, viruses and fungi. It is also used to view bacterial flagella and plasmids. It is used to view the shape and size of microbial cell organelles. And it is also used in nanotechnology, okay, that is to study nanoparticles such as zinc oxide nanoparticles. And then it is used to study and differentiate between plant and animal cells, okay. So these are the main applications of TEM. So this is all about transmission electron microscope. Thank you.